Hello, once again, welcome back to Lab. Let's review the structures of the mammalian brain. So I have a bunch of wet brain specimens here from all of the cats dissected in this laboratory. And so we start first with the undissected or just the superficial structures of the cat's brain, and then maybe we'll go to the structures that can be seen in our plastic model specimen located there, containing the detailed structures of the mammalian brain that cannot be seen in our wet specimens. So let's start with the dorsal aspect. So first and foremost, you'll see that the brain will be covered by tough connective tissue that in life actually encases a cavity inside filled with fluid. So those tissue coverings, connective tissue coverings of the brain would be the meninges. And as you know, the mammalian brain is covered by 3 layers of connective tissues or meninges. The outermost and the toughest one would be the dura mater. This is the dura mater, and here you'll see that the dura mater has been removed, and the other 2 layers, the 2 deepest layers or meninges, are closely applied to the brain substance itself, and it's impractical to dissociate them, but those white stuff in between these convolutions of the brain, the gyrus, would be the arachnoid matter, the middle meningeal layer, and then the pia mater, the 3rd and deepest meningeal layer, would be the one that's actually close or almost fused to the brain substance itself. So it's impractical to dissociate the 2. But suffice it to know that this one, the most obvious and easily detachable layer, is the dura mater. The second would be the arachnoid mater, and the 3rd and deepest meningeal layer is the pia mater, containing lots of blood vessels. It's highly vascularized, so it supplies, one of its function of the pia mater would be to supply blood nutrition to the brain, which is a highly active organ. So as I said, there are cavities located within or in between the meningeal layers, and the one directly deep to the dura mater is called the subdural space, and the other one would be the subarachnoid space, but we won't be able to see it. But here, those spaces, like the subdural space, would be in life filled with cerebrospinal fluid, which acts like a cushion, allows the brain to float in that fluid, acts as a cushioning mechanism so that the brain will not be physically injured as you shake your head, for example. Right? And also the cerebrospinal fluid carries a lot of the metabolic waste products of the brain and flushes it and drains it into the lymphatic system. So it's connected to the lymphatic system. Right, so let's examine the dorsal structures first. In the mammalian brain, the most prominent structures that you'll see are these 2 parts here called the cerebral hemispheres. You have the left and the right cerebral hemispheres. And the cerebral hemispheres of mammals are characteristically convoluted, kind of like intestines, right? And those convolutions are called gyri, or singular gyrus. And those shallow grooves in between the gyri are called sulci, singular sulcus. And this groove here, deep groove separating the left and right cerebral hemispheres, would be the longitudinal cerebral fissure. The other prominent structure in the mammalian brain would be the cerebellum, literally the little brain, cerebellum. So if you reflect the cerebral hemispheres and look a little bit deeper, you'll see 4 circular structures there. So this entire structure would be part of the midbrain, called the corpora quadrigemina. Specifically, those 2 anterior globes there are the superior colliculi, involved in visual reflexes, and the posterior 2 globular structures there are the inferior colliculi, involved in auditory reflexes. And there should be, we'll look at a better brain, there should be a little knob structure unpaired there. There, you see there. So this would be your superior colliculi, but deeper there is part of the epithalamus, okay, called the pineal body or the pineal gland, which secretes a hormone called melatonin, which regulates body clock, the circadian rhythm of the animal. So it's a pineal body. And so cerebral hemispheres, cerebellum, this one, the hind portion of the brain, is the medulla oblongata, which indistinguishably transitions into the spinal cord. But this one, this more cranial expanded portion, it would be the medulla oblongata. Let's examine the ventral. In the rostral portion, you'll see 2 structures called the olfactory bulbs. So in this specimen, it has been removed already, but there should be 2 olfactory bulbs here, like this one, olfactory bulb. And the olfactory bulbs will receive olfactory sensations from the olfactory nerve and deliver it via the olfactory tracts into a portion of the cerebral hemisphere called the piriform lobe. There's a special portion of the cerebral hemisphere that processes olfactory information, smell information. And that groove that delineates the piriform lobe from the rest of the cerebral hemisphere would be the rhinal fissure. Alright, so that would be the olfactory system. And next, you'll see here, if you move more posteriorly, you'll see part of the hypothalamus. So this region here would correspond to the hypothalamus, which has many subparts. So this one, this X-like structure, is the optic chiasma. It's the point of decussation or crossing of the 2 olfactory nerves. So here's another brain which has a better view of the optic chiasma. And here would be the truncated portion of the optic nerves, and then these would be the optic tracts. And this region, as I said, is the hypothalamus. And here you see a hole. In life, that there should be a little sac-like structure there. That was the position of the pituitary gland, specifically the posterior lobe of the pituitary gland. which was suspended via an infundibular stalk. And this rounded eminence or rounded portion here at the base of the pituitary gland would be the tuber cinereum. And then if you move more posteriorly, so here, the hole, that's actually an entrance. If you insert a probe, you'll be probing into the third ventricle of the brain. So optic chiasma, tuber cinereum, missing is the pituitary gland. And then if you move more posteriorly, there's another bulge corresponding to the mammillary bodies. Mammillary bodies posterior to the tuber cinereum. Mammillary bodies. And then these two here are the cerebral peduncles, already part of the midbrain, not part of the hypothalamus. And this one is not part of the midbrain anymore, that's part of the hindbrain, specifically the metencephalon. So that would be the pons. Another part of the metencephalon would be the cerebellum. And the cerebellum has a median worm-like structure called the vermis, literally the worm of the cerebellum. And the two lateral bulges of the cerebellum would correspond to its hemispheres, the cerebellar hemispheres. So there's a worm or vermis of the cerebellum and then the cerebellar hemispheres. Alright, and this is the pons. And then if you move more posteriorly, you'll reach the part of the hindbrain called the myencephalon. And the myencephalon consists of the medulla oblongata, which has subparts. So these lateral bulges here, posterior to the pons, would be the trapezoid bodies. And this longitudinal bands here are the pyramids or the somatic motor tracts. Motor tracts carrying motor impulses from the higher brain regions down to the spinal cord to control the skeletal-muscular system, for example. Somatic motor tracts or pyramids, and then this would be the spinal cord already. Now let's look at the mid-sagittal section of the brain to look at deeper structures. So this would be the cerebral hemisphere, specifically the left cerebral hemisphere. And the two hemispheres are actually linked via a transverse band of connective tissue there, as you can see. In the median sagittal or mid-sagittal section, it's this structure here called the corpus callosum. So that's a corpus callosum. And the anterior end of the corpus callosum is called the genu. The posterior end would be the splenium. And you see here, if you perfectly cut the brain mid-sagittally, there's a covering here called the septum pellucidum. But if it's not perfect, you'll see inside the cavity of the lateral ventricle. So there are 4 ventricles or cavities in the brain, the 2 lateral ventricles, and then the third ventricle here. So the lateral ventricle is inside there, covered by the septum pellucidum. This is the third ventricle, located in the diencephalon. And then the fourth ventricle here is located in the hindbrain, the fourth ventricle. And in between the fourth and the third ventricle is the cerebral aqueduct, located in the midbrain. So let's go back to the mid-sagittal section. So this one here is called, also has a name called the fornix. Fornix. That ventral anteriorly projecting structure, the fornix. Right, and this region here would be the diencephalon, which has three portions. The more dorsal one would be the epithalamus, the middle one would be the thalamus, and the ventral most portion of the diencephalon would be the hypothalamus, whose structures we have examined here in the ventral aspect of the brain. So this rounded structure here is part of the thalamus, called the massa intermedia, or intermediate mass. So the thalamus is the part of the brain where a bunch of sensory signals are relayed into for further transmission to the higher portions of the brain. So it's like a sensory hub, the thalamus. And what else? So this region here would be the midbrain already. These are actually the corpora quadrigemina, right? The superior colliculus and the inferior colliculus. And this is the cerebral aqueduct. And the floor of the midbrain is called the tegmentum. The cerebral peduncle is also part of the midbrain. Let me show you again. Cerebral peduncles. Cerebral peduncle. The floor of the midbrain is generally called the tegmentum. This area here, tegmentum. And this would be the corpora quadrigemina. And this would be the pons, cerebellum, and then trapezoid body. You see the median sagittal section of the cerebellum reveals its gray matter called the cerebellar cortex, and then the white middle portion would be its white matter. But it has a fancy name because of its tree-like appearance. The white matter of the cerebellum is called arbor vitae, literally tree of life. The cerebral hemisphere also has gray and white matter, but it's not obvious in this preserved specimen. It has been discolored, but the outer layer should be grayish in color. That's the cerebral cortex, made of gray matter, and then the middle portion would be white. That's the white matter. So the difference between gray and white matter is the abundance of cell bodies of neurons. So the cell bodies would be more concentrated in the gray matter areas, whereas the white matter is white because they contain the axons, which are covered by a fatty layer of myelin sheath. That's why they're white, because of all of the fat, white. So gray matter, cell bodies; white matter, axons covered by myelin sheath. So here's a close-up of the hindbrain. This region would be the medulla oblongata, and this one would be the cerebellum. And I just want to point out those longitudinal tracts you see in the medulla oblongata, dorsal portion. Those have names. Those are the somatic sensory columns of the medulla, containing axonal fibers, sensory fibers, relaying information from the body to the brain. And this, specifically these median longitudinal tracts here, are the clava, and lateral to it would be the tuberculum cuneatum. So both are somatic sensory columns. Clava, tuberculum cuneatum. So in general, the arrangement of fibers in their central nervous system is that dorsally you'll find the sensory tracts like this structures, the clava and tuberculum cuneatum. And then on the ventral side, you'll see the motor tracts relaying information from the brain to the rest of the body to control the muscles, for example. And we have already seen the pyramids, right? The pyramids of the medulla oblongata, also known as the somatic motor tracts. The pyramids. So motor, ventral, dorsal, sensory. And those would be the clear structures that you can see from this wet specimen of the cat. Let's turn to the plastic models.